we're going to discuss cell signaling with linked protein receptors. And as they're called, it's G-protein coupled receptors rather than linked receptors. I believe it is good to have an understanding of what cell signaling is, what G-protein is, and what the GPCR or the G-protein coupled receptor may mean. Cell signaling is considered to be any communication that controls basic cellular activities. It's the cell's ability to perceive and respond to the perception of its microenvironment that can involve development, tissue repair, immunity, and so on and so forth. In this picture, I have shown cell sending signals, and the signal is usually transmitted in terms of a ligand or ligand. It could be ions, other molecules, and it can be a perception of audio, visual, and hormones, and it works on a cell that is called target cell if it has a receptor to receive that signal. If it does not have a receptor to receive that signal, then it would be called a non-target cell. Following receiving the signal, it may produce a response. Each receptor can bind to one or few ligands. So they're specific. Only certain ligands can bind to certain receptors with a lock and key mechanism. So to elicit response, the receptor passes on the signal to various types of proteins called G-proteins. I have shown a cell membrane. We'll call this side as the outside of the cell or the cell membrane, and this being the inside, where I showed a receptor, a ligand, in this case a hormone, that is supposed to attach to this receptor. Following that, it will elicit some form of conformational change that will make some impact on the G-protein. The G-protein acts on some other protein that may be present in the membrane to elicit the response. At the receptor are usually transmembrane, passes from inside of the cell to the outside. And of course, it has to have some portion outside the cell in order for it to receive the signal that is coming from the outside the cell. And of course, it has to have some portion within the cell or inside the membrane so that it can elicit the response that may be present within the cell. Let's take a look at the G-protein. It's present partially inside the membrane and partially within the cell. And of course, that is working on, in this case, enzyme, which happens to be another protein, is present inside or within the membrane. Let's talk about what are G-proteins for the cell signaling. It acts as a molecular switch inside the cell. It transmits the signal from a variety of stimuli outside the cell to its interior. Since these proteins bind to the purine nucleotide, guanosine, in the form of a guanosine diphosphate or guanosine triphosphate, because it attaches to guanosine triphosphate, the G from the guanosine linked to this protein, and hence the name G-protein. It is important to have some understanding of the structure of the guanosine triphosphate to understand how it attaches to the protein called G-protein and what happens in terms of chemistry to understand how that may be involved in cell signaling. There are two distinct families of G-proteins. One kind is called large G-proteins. The other kind is called small G-proteins. The large and small size are coming from the fact how many subunits they may have. For the large protein, it has alpha, beta, and gamma, all those three subunits, and therefore they're called heterotrimeric. That means three different kinds, subunits. The smaller ones have only one subunit. Here I have shown the different subunits of the larger G-proteins. Here I have shown that there is an alpha subunit and there are beta and gamma subunits together called G-protein. Alpha and gamma subunits are drawn in a different way rather than the circle for beta subunit. And that just happens to be the 3D structure of different subunits. Particular portions that are attached to the inside of the cell membrane are the portions called lipid anchors. Now, Gprotein is generally in its inactive form. It needs to be activated in order for it to be functional. The activation takes place in 2 steps. Guanosine diphosphate, it attaches to the alpha subunit of the Gprotein. The second step is activated Gprotein then attaches itself to guanosine triphosphate. At the receptor stretches itself from outside to inside. There are 7 transmembrane alpha helices. And here I have shown that in terms of going down 1, 2, 3, 4, 5, 6, and 7. GPCR or Gprotein coupled receptors are also called 7 transmembrane receptors. And receptor has a specific region to receive the ligand. So specific ligand can attach to the specific binding site of these receptors. There are a few things about GPCRs or the receptors that we must keep in mind. That is, they are only found in eukaryotes. They are unique membrane receptors. They are, to this date, the largest known class of membrane receptors. And just it happens so the functions of many Gprotein as well as their receptors nearby or the GPCRs are yet unknown. And because of that, they are studied on. As a matter of fact, in 2012, the Nobel Prize in Chemistry was given out for the study of Gprotein coupled receptors involved in cell signaling. To our knowledge, human has over 1,000 known various types of Gprotein coupled receptors. GPCRs are target for about 50% of the common modern medicinal drugs. Regulate the immune system, they regulate the growth, smell, taste, vision, sight, behavior, mood in terms of serotonin and dopamine, and the list goes on and on. How the actual cell signaling takes place. Since now we have some understanding of the cellular microenvironment being outside the plasma membrane to the inside of the plasma membrane, we have a little bit of idea what Gprotein structurally and chemically may look like, and what Gprotein receptors may structurally look like, and how the signal may come from outside in the form of a ligand. Here it is shown by a green dot, and how the signal may go to the inside of the cell. The ligand here is binding to the receptor. And then what happens is the GPCR undergoes a conformational change. When there is a conformational change on the receptor due to ligand attaching to the receptor, that makes an impact on the Gprotein to dissociate or separate out the alpha subunit from the beta or gamma subunit. The guanosine diphosphate on the alpha subunit, and it is replaced by guanosine triphosphate. If GDP is replaced by GTP, then it helps the alpha subunit to separate out from the beta and gamma subunit. All this beta and gamma subunit are dimer, so that gets dissociated out of that whole Gprotein unit, making the alpha subunit separate. Both this GTP associated alpha subunit and the beta and gamma dimer can separately take part in transmitting the signal. This alpha subunit works on some target protein inside the cellular membrane. So for our purpose, we'll call that target protein maybe an enzyme that will carry out certain reactions within the cell. As long as this ligand is attached to the receptor, this signal keeps on transmitting and keeps on making the message to do its work within the cellular environment. When the GTP loses a phosphate through a reaction called hydrolysis, and goes back from GTP to GDP, the reverse of the sequence happens. The signal stops and the whole thing goes back where it was in the first place.